in your next period, you are going to undertake a first-hand investigation, a practical, to investigate some of the roles of chemical reactions in the digestion of our food. We need to talk a little bit about the background for this prac before the meaning of it's going to make any sense at all. Digestion, recall from previous years, is a process of breaking food into nutrients that your body can absorb and use. There are two types of digestion. There's mechanical digestion that involves breaking the lumps of food up into smaller pieces and then mixing them together, stirring them together. So chewing and swallowing is an example of mechanical digestion. The way that your stomach churns and the muscle contractions that it goes through, that's another example of mechanical digestion. But there is also chemical digestion, and we've never discussed that. Chemical digestion is all of the chemical reactions that your body conducts to break those big food molecules into smaller ones. That's because the molecules in your food are for the most part much too large to cross your cell membranes, so they can't be used by the body until they've been broken down into smaller molecules that can fit into the cell. So imagine the cell membrane being like a farm gate. It's big enough to allow a small vehicle to pass through, like a car or a four-wheel drive or a ute. But it's not big enough to allow a really big vehicle to pass through. And so the same with your cell membranes. They do have small holes in them where fairly small molecules can pass through. But the really big molecules that mostly make up our food get stuck trying to pass through those gates in the cell membrane. So that means that chemical digestion is breaking large molecules into molecules that are small enough to pass into our cells. The body uses helper chemicals that are called enzymes that allow chemical digestion to occur. So, with sugar, for example, in order to get the energy out of sugar, your body uses these chemicals that are called enzymes, and the enzymes don't actually participate in the chemical reaction themselves. They just allow that chemical reaction to occur very quickly at a very, very low temperature. The same chemical reaction happening in a jar on a bench would have to be heated to hundreds of degrees Celsius. But of course, in your body, they have to happen at 37 degrees Celsius. And that's what the enzymes do. They allow the chemical digestion to occur more quickly and at a lower temperature. For example, starch is a very large molecule that's made of many, many small sugar molecules. Amylase is the enzyme that helps to break starch into sugar. So if you combine starch with amylase, it will get broken up into the sugar molecules, and those sugar molecules are then finally small enough to pass into the cells in your body. Proteins are also very large molecules. They are made by smaller molecules called amino acids. Again, the protein is too large to pass into this across the cell membrane into the cell, but the amino acids are small enough to. Pepsin is the enzyme that helps to break the proteins into amino acids. So our aim in this investigation is to demonstrate the role of amylase in the digestion of starch and the role of pepsin in the digestion of proteins. So there are two methods. The first method is to investigate the role of amylase. Begin by labeling 3 test tubes. One of those test tubes will say water, another one will say amylase, and another one will say amylase and hydrochloric acid. Then add a Nutrios baby snack, you'll find that in your lab, to each of your 3 test tubes, 1 baby snack per test tube. Then cover the baby snack with water, or cover it with amylase, or cover it with amylase mixed with acid, according to the labels that you've recorded on your test tubes. Put them in a test tube rack and leave them overnight. The next day, shake your test tubes very well. Use a plastic pipette to transfer about 40 drops of the solution from your first test tube, that means your water test tube, into a new, clean, dry test tube. Add 10 drops of Benedict's solution to that new test tube and shake to mix. Then heat very lightly over a Bunsen burner. I say very lightly. You only need this reaction to be heated to about 50 degrees. So there's no point cranking it up onto that blue heating flame and leaving it there for 2 minutes. You're just going to boil off all your solution. The mixture only needs to go to about 50 degrees. So it needs to be warm, maybe even hot, but not boiling. A long, long way from boiling. Then repeat step 4, that's the last step, for both the amylase test tube and the amylase and acid test tube. If the Benedict's solution turned yellow, green, or brick red, then glucose was present. And glucose is a form of simple sugar that's small enough to cross into the cells of your body. So what we're testing for is, overnight, has the starch in our baby snack been broken up into glucose, which is a form of sugar. All right, your second method is to investigate the role of pepsin. Again, label 3 test tubes. The first one is called water, the second one is called pepsin, and the third one is called pepsin and hydrochloric acid. Add a very small amount of meat to each of your test tubes. About the size of your fingernail will do. In fact, even the size of your smallest fingernail will probably be enough. Cover that meat with water or with pepsin and acid, again, according to your labels. Leave your test tubes in a labeled test tube rack overnight beside your amylase test tubes. All right, risk assessment. First of all, no secrets here. First of all, the Bunsen burner is a naked flame. Also, the test tubes that you heat over Bunsen burners might bump. That means boil in a flash, literally in an instant. And that can splash hot solution on you, so it's pretty dangerous. Acid is corrosive, and baby snacks and meat may be contaminated by heaven knows what. Therefore, girls, you need to tie your hair well back. Boys, you'll wear an apron to secure your ties, and you must never turn your back on a Bunsen burner. Always heat your test tubes at an angle pointed away from you. Keep them moving through the flame, and remember, those test tubes that you're heating only need to go to about 50 degrees Celsius. Wear your safety glasses at all times. And never eat anything in the lab, even if it looks like food and is called baby snacks. The minute it's come into the lab, it's not food any longer. All right, we'll see you in the lab tomorrow.